We now need to turn our attention to the autonomic nervous system. So the first thing we want to address is what is the fundamental difference between the somatic motor system and the autonomic motor system. Now, many people will answer that question by saying that the somatic nervous system is voluntary and the autonomic nervous system is involuntary. But that's not correct. That's not true. The somatic nervous system can act voluntarily or involuntarily. The autonomic nervous system, while it usually acts involuntarily, can also act voluntarily. So that's not the correct distinction. The correct distinction is based upon what kind of end organ is being innervated. The somatic nervous system innervates skeletal muscle. That's always true. All somatic motor nerves innervate skeletal muscle. All skeletal muscles are innervated by somatic motor nerves. In contrast, autonomic motor nerves, the autonomic system innervates smooth muscle, cardiac muscle, and glands. That's always true. So that is the true distinction between the two. Now, there is also a difference in terms of the pathways used by these nerves to innervate those end organs. More specifically, what we can say is that the somatic nervous system innervates its skeletal muscle by way of what is called a one-neuron pathway, meaning that from the central nervous system out to that skeletal muscle, the pathway is composed of a single neuron. It may be a neuron with a very long axon if the skeletal muscle is far away from the spinal cord or brain, but nonetheless it's still one neuron. In contrast, the pathway to innervate smooth muscle, cardiac muscle, and glands by the autonomics is a two-neuron pathway, in which case the first neuron leaves the central nervous system. It doesn't innervate the end organ, but rather it innervates the second neuron. The second neuron then innervates the end organ. We said that in the case of the somatic, the cell body of that first neuron is in the central nervous system. It might be in the brain, it might be in the spinal cord, but it's in the central nervous system. In the case of the autonomic nervous system, the first neuron is also in the CNS. Maybe brain, maybe spinal cord, but it's in the CNS. And the second neuron is always in the peripheral nervous system. So the first neuron leaves the CNS, enters the PNS, and synapses on the second neuron in the PNS. Since it's in the PNS, we would call those cell bodies, we would say those cell bodies are in ganglia. Because we know the location of the cell body, that means we know the embryonic origin of those cells. Since the somatic motor nerves have their cell bodies in the CNS, that tells us that they are derived from neurolectoderm. Since in the autonomic nervous system we have one cell in the cell body in the CNS and the other in the PNS, that means the first neuron comes from neurolectoderm and the second neuron comes from neural crest. So when we look at this schematically, this could be a representative somatic motor neuron. We would have a cell body. It would be in the spinal cord or in the brainstem. It would have an axon that would proceed all the way out through the peripheral nervous system, however far it has to go to get to a skeletal muscle that would be out here, and it would innervate it. It would be one neuron the whole way. In the case of the autonomic nervous system, the first neuron has its cell body in the CNS. The second neuron has its cell body in the peripheral nervous system in a ganglion. The first neuron is innervating the second neuron is innervating the end organ. Because this second neuron cell body is in the ganglion, the axon going to it is called preganglionic. So the first neuron's axon is called a preganglionic fiber. The second axon is called postganglionic. That's what's going from the ganglion to the end organ. So in the case of the autonomics, we always have this two-neuron pathway, with the first neuron innervating the second neuron innervating the end organ. There is only one exception to that, and that exception is the innervation of the adrenal medulla. The adrenal medulla is innervated by a one-neuron pathway. In other words, this neuron would be innervating the adrenal medulla. This neuron wouldn't exist. The reason for that is that, as we've already said, these second neurons are derived from neural crest. Well, the adrenal medulla is derived from neural crest. So if we sort of substituted adrenal medulla for this oval, eliminated this axon, since those cells don't have axons, we could then say that this would be the adrenal medulla. It's being innervated by this. So it's innervated by a one neuron pathway. So the reason the adrenal medulla is different is based on its embryology, that it is derived from the same cell types, neural crest cells, as are derived the postganglionic neurons of the system. So it itself is the postganglionic in a developmental sense. So that describes the generalities of the autonomic nervous system. What we're now going to want to do is distinguish two parts of the autonomic nervous system, the sympathetic nervous system and the parasympathetic nervous system. We're going to compare the two portions of the autonomic nervous system, the sympathetic portion and the parasympathetic portion. And before we talk about the differences between those two portions, let's make sure we understand the similarities. Since both the sympathetic and the parasympathetic nervous systems are parts of the autonomic nervous system, everything that we said that was true about the autonomics has to be true about both sympathetics and parasympathetics. That is to say, both sympathetics and parasympathetics innervate smooth muscle and cardiac muscle and glands. Both sympathetics and parasympathetics do that by way of a two-neuron pathway with the first neuron cell body being in the central nervous system and being derived from neural ectoderm, and the second neuron cell body being in the peripheral nervous system and being derived from neural crest. So all of those things are the same. So what are the differences? Well, we can start off by sort of digging a little deeper and asking some more specific questions. Let's talk about the preganglionics. We said that the preganglionics have their cell bodies in the central nervous system. But if we ask the question, where in the central nervous system, then we'll get two different answers. In the case of the sympathetic nervous system, we can see on this slide on the left that the cell bodies of the preganglionics in the sympathetic nervous system are all found in the spinal cord and are found at spinal cord levels T1 to L2. We notice that there are no cell bodies above T1 in the spinal cord or the brainstem, and there are no cell bodies of the sympathetics in the spinal cord below L2. On the other hand, for the parasympathetics, if we look to see where the cell bodies of their preganglionics are found, on this slide on the right, we then see that they are not found at levels T1 to L2, but rather they are found at higher levels in the brainstem associated with cranial nerves 3, 7, 9, and 10, and at lower levels in the spinal cord at sacral levels associated with sacral 2, 3, and 4. So for sympathetics, the preganglionic cell bodies are from levels T1 to L2. For parasympathetics, they're at cranial and sacral levels. And that's why the sympathetic outflow is often called the thoracolumbar outflow, and the parasympathetic outflow is often called the craniosacral outflow. We said that for both sympathetics and parasympathetics, the postganglionic cell body is in the peripheral nervous system. So we can ask the same question, where in the peripheral nervous system? And so we need a way of defining regions of the peripheral nervous system. And there are fundamentally two ways we can do it. We can refer to regions of the peripheral nervous system based on their relationship to the central nervous system. In other words, how far away or close to the central nervous system are we? Or, alternatively, we can do it based upon its position relative to the end organ out here. In other words, how far or close to the end organ is that cell body? And so we understand that it's sort of saying the same thing in two different ways. If the cell body is close to the central nervous system, that means it's far from the end organ. If it's far from the central nervous system, it means it's close to the end organ. And as it turns out, in the case of the sympathetics, the postganglionic cell bodies are found close to the central nervous system, which means they are far from the end organ. And in the case of the parasympathetic, they are far from the central nervous system, which means they are close to the end organ. Based upon that difference, we can say something about the length of the axons. For the sympathetic, since the preganglionic axon does not have to go very far because the postganglionic is close to the CNS, it's going to have a short preganglionic axon. But since that cell is far away from the end organ, it's going to have a long postganglionic axon. So sympathetics have short preganglionics and long postganglionics. And in contrast, since the parasympathetic postganglionic cells are far from the CNS but close to the end organ, therefore they will have long preganglionics to get to those distant postganglionics, but very short postganglionics because these cells are very close to the end organ. So those are the fundamental differences. There are also differences in terms of function. Not so easy to characterize. As a generalization, we can say that sympathetics tend to control responses that fit into the fight or flight response. In other words, the things we do in an emergency, like increase heart rate and sweat and dilate our airways. And parasympathetics tend to relate to the functions that sometimes are referred to as the rest and digest functions, slowing down of the heart rate, increasing peristalsis, increasing digestive secretions, and so forth. And that's a generalization, not true in every single instance. But we can generalize and say that generally the functions have opposite outcomes. So, for example, if sympathetics dilate the pupil, parasympathetics will constrict the pupil. If sympathetics increase heart rate, parasympathetics will decrease heart rate, and so forth. Now let's talk about the specifics of how these nerve fibers get to where they have to go. What are the specific pathways? And let's start our discussion with the sympathetic nervous system. So the sympathetic nervous system has one fundamental issue that it has to deal with when we consider its pathways. And the issue is this. Even though the preganglionic fibers arise from a fairly restricted region of the nervous system, only from T1 to L2 levels, nonetheless, the distribution of the sympathetic nervous system is to the entire body. All parts of the body get sympathetic innervation. One example that we can use to illustrate that point is that sweat glands, for example, are only sympathetically innervated. And certainly we have sweat glands all over our body, going from the top of our scalps all the way down to the soles of our feet. So we have to be able to get sympathetics to all parts of the body, even though we start from a fairly limited region of this central nervous system. So how are we going to do that? Well, let's start off with the simplest of the issues, and that would be, how would we go about innervating structures that are at the T1 to L2 level? In other words, in the trunk. Let's say we wanted to innervate a sweat gland on the chest wall or a sweat gland on the abdominal wall, either anterior wall or posterior wall. How would we do that? Well, we would start from a preganglionic somewhere between T1 and L2. That preganglionic would send its axon out to the postganglionic. Remember, postganglionics for the sympathetic system are close to the CNS, and here we see them found in the sympathetic chain, which is found right outside of the vertebral column. So we have these short preganglionics going to the sympathetic chain at that same level, synapsing on the postganglionic, and then the postganglionic continues on out to the periphery at that level and innervates. And this little star here, or asterisk, represents cutaneous structures such as sweat glands or smooth muscle of hair follicles and so forth. So it's going to innervate structures at that same level. So if we want to innervate a sweat gland that's, let's say, in dermatome T8, we would start at the T8 preganglionic, go to the T8 sympathetic ganglion, and then distribute out with the T8 spinal nerve to the sweat gland at the T8 level. To take a look at the specifics of how that happens, let's take a look at this slide. Here we see the preganglionic. It's in the central nervous system, somewhere between T1 and L2. It's in the gray matter. But in contrast to the somatic motor fibers that have their cell bodies in the anterior horn of the gray matter, these sympathetics have their cell bodies in the intermediolateral horn of the gray matter between T1 and L2. But nonetheless, they still exit through the ventral root since they are motor fibers. They go out through the ventral root into the spinal nerve. And now remember, they have to get to a postganglionic. They're heading to the postganglionic in the sympathetic chain ganglion, which we can see over here. So we need a way to get from the spinal nerve to the sympathetic ganglion. And what we have here is a branch that communicates between spinal nerve and sympathetic ganglion, and that's called the white ramus communicans. Ramus means branch, communicans means it communicates. White because it looks white, because these nerve fibers, these preganglionic nerve fibers, are heavily myelinated. Myelin looks white, so this branch looks white. So it uses the white ramus communicans to get from the spinal nerve to the sympathetic ganglion. When it gets to the ganglion, remember what's found in the ganglion is cell bodies, postganglionic cell bodies, and so it synapses on a postganglionic neuron. So now the postganglionic neuron has to get to the end organ to innervate it. So how are we going to accomplish that? The postganglionic cell body is in the sympathetic ganglion. The end organ is out here someplace, so it needs a way of getting from here to there. And what we have running close by, of course, is the spinal nerve and its branches, the dorsal and ventral ramus. So if it can just get to a spinal nerve, it would be able to simply tag along, sort of become incorporated with everything else of the spinal nerve and travel with it. So that's what's going to happen. It gets from the ganglion to the spinal nerve, again by a communicating branch, ramus communicans, but this time, because it's made of postganglionic axons, which are unmyelinated, this looks gray, so we call this the gray ramus communicans. So the postganglionic gets from the ganglion to the spinal nerve with the gray ramus communicans, joins the spinal nerve, and then either travels with the ventral ramus or travels with the ramus. These fibers are also postganglionic here or here, either way. Travels with it to wherever it goes, and then when it gets there, it innervates an end organ like a sweat gland or a smooth muscle cell. So that's pretty straightforward. That's how we innervate structures in the body wall at levels T1 to L2. But what if we want to get below L2 or above T1? Let's say we want to get to a sweat gland in the lower limb, or if we want to get to a sweat gland on the neck or the back of the scalp. How are we going to do that? So to get below L2, what we're going to do is we start as close as we can. So we'll start from a preganglionic at the L2, maybe the L1 level, let's say the L2 level. The preganglionic goes into the ganglion, again using the white ramus to get there. Even though there are cell bodies that are in that ganglion, that's not where they synapse. They continue down the sympathetic chain to a lower ganglion. So if this is the L2 ganglion, let's imagine there's an L3 ganglion here, there's an L4 ganglion here, there's an L5 ganglion here, and so forth all the way down to the bottom of the sacrum. And so let's say it goes to the L4 or maybe the L5 ganglion, and it synapses there. So even though these preganglionics coming from L2 enter the L2 sympathetic ganglion, they don't synapse in the L2 sympathetic ganglion. They descend the chain to a lower ganglion, L3, L4, L5 or something, and synapse in that ganglion. Having synapsed there, then the postganglionic has to get from that ganglion out to the periphery, so it uses the spinal nerve at that level. So if it's the L4 ganglion, the postganglionic will travel with the L4 spinal nerve. If it's the L5 ganglion, it will travel with the L5 spinal nerve, and so forth. Follow that spinal nerve to wherever it goes, and so it will get to a sweat gland in the L4 or the L5 dermatome or whatever level we're at. That's how we manage to get below the L2 level. We use exactly the same process, but in reverse, to get above the T1 level. So if we want to get to a sweat gland up in the cervical region, we would start as high as we can, which would be at the T1 level. The preganglionic would enter the T1 ganglion. Again, even though there are cell bodies there, it doesn't synapse there. It ascends to one of the higher ganglia, one of the cervical ganglia. It synapses in that cervical ganglion. The postganglionic then has to get from that ganglion to the periphery using the spinal nerve at that level. So it will travel with the C3 or C4 or C5, whatever level it's at, travel with that spinal nerve to get to those dermatomes to innervate the structures in the body wall at those levels. So, we've now managed to innervate body wall structures all the way from the cervical levels down to the sacral levels. We still have some other structures that need to be innervated, and those are structures that are visceral structures within the body cavities, within the abdomen and pelvis, for example. The way we do that is a little bit different. We take preganglionics from the thoracic or lumbar region, typically from T5 on down. Those preganglionics leave the spinal cord by way of a white ramus, but when they get to the sympathetic ganglion, they don't synapse there. They also don't ascend or descend through the chain, but rather they continue right through the ganglion and come out and continue out into the periphery. They still have not synapsed, so they are still preganglionics and they continue out into the periphery. Now we know that they can't go all the way to the end organ to innervate it because preganglionics don't do that. They have to synapse on postganglionics. And since we passed up the opportunity to synapse on a postganglionic in the sympathetic chain ganglia, we need to have a second chance, so to speak, other ganglia further out in the periphery. So we have a series of ganglia that are either called collateral ganglia or, as labeled here, prevertebral ganglia, or they can also be called preaortic ganglia. They all mean the same thing. And these preganglionics synapse on those prevertebral or preaortic ganglia. Now, those ganglia are found in the abdomen on the posterior abdominal wall, specifically right on the front surface of the abdominal aorta. So that's why they're called either prevertebral or preaortic. And those preaortic ganglia that are on the anterior wall of the aorta are not just randomly along the aorta. They are found in groups where the major arteries come off the abdominal aorta, and the ganglion takes its name according to which artery it's found alongside. So the ganglion that's near the celiac trunk will be called the celiac ganglion. The artery that's near the superior mesenteric artery will be called the superior mesenteric ganglion, and so forth. So the preganglionic that did not synapse in the sympathetic chain continues out to one of these preaortic ganglia and synapses there. Those nerve fibers collectively are referred to as sympathetic splanchnic nerves. So sympathetic splanchnic nerves, whether from thoracic levels or L1 and L2 lumbar levels, those sympathetic splanchnics are preganglionic sympathetics that pass through the sympathetic chain without synapsing and continue on out to a preaortic ganglion or prevertebral ganglion and synapse there. The postganglionics now need to get to an end organ to innervate the end organ. Now we know that the postganglionics that were in the sympathetic chain, when they needed to get to an end organ, used the spinal nerve as their method of transportation, so to speak. They tagged along with the spinal nerve, and that's how they got to their end organs. Well, these postganglionics that are in the preaortic ganglion don't have spinal nerves running near them, so they can't do that. Instead, what they have are those very arteries that are the branches of the aorta, so they can hop on the artery and travel with the artery. They run along the surface of the artery. So postganglionics from the celiac ganglion will run along the surface of the branches of the celiac trunk. Postganglionics from the superior mesenteric ganglion will run along the surface of the branches of the superior mesenteric artery. Wherever those arteries go, the postganglionics go with them. They eventually lead to some organ in the body, in the abdomen, and they innervate the end organs in that organ. So smooth muscle cells or glands in those organs. So they're using the branches of the abdominal aorta as their pathway to get to their end organs. So using one or another of the pathways we've just described, we've now managed to almost solve the problem. The problem was we have a limited origin, but we have to distribute all over the body, and we've managed now to get from the top of the head to the bottom of the feet and to the organs within the body cavities. There's one place left that we haven't covered, and that is the front of the head. The front of the head doesn't receive any spinal nerve innervation. It receives cranial nerve innervation, so we can't use spinal nerves to get there. And certainly the front of the head doesn't get branches of the abdominal aorta. So we need another method. And we have a method that's sort of a combination of the methods we've already talked about. Since we want to get to the head, we start as close as we can, which would be the T1 level. We go into the T1 ganglion with our preganglionic using the white ramus to get there. We don't synapse there, rather we ascend through the sympathetic chain and we go as high as we can. Since we want to get to the head, we go to the very top of the neck. That's the superior cervical ganglion. That's the last chance to synapse, so that's where it synapses. The postganglionic now has to get from that ganglion to end organs in the head, like sweat glands or smooth muscle. So we know that in the abdomen it used branches of the abdominal aorta to get to end organs. We don't have the abdominal aorta in this region, but we do have the external and internal carotid arteries, and the carotid passes right past the superior cervical ganglion. So these postganglionics hop onto the carotid system, internal and external. They travel along the branches of the carotids, and wherever those branches go, these sympathetics go with it. So since the whole front of the head obviously has a blood supply, we now have a pathway to bring sympathetic innervation to the head. Now, recognize that that pathway to get sympathetics to the head is a fairly long pathway. It starts here in the thorax and then has to ascend all through the cervical sympathetic chain and then all along the carotid arterial system. When you have a long pathway like that, an interruption in the pathway anywhere along its length will interrupt sympathetic innervation or nerve innervation to the end organ. When we lose sympathetic innervation to the head, there are a few easily observed findings associated with that. And that would be a loss of the innervation to the smooth muscle that elevates the eyelid, so that results in a drooping of the eyelid. A loss of the innervation to the smooth muscle that dilates the pupil, so that would result in a constriction of the pupil. And a loss of the innervation to the sweat glands, so that would result in a loss of sweating. Those three easily observed clinical findings are collectively given the name Horner's syndrome. So that means that any interruption anywhere along the pathway to the sympathetic innervation to the head will result in a Horner's syndrome. So when a patient displays a Horner's syndrome, that tells you that there's an interruption in the pathway of the sympathetics to the head. What it does not tell you is where that lesion is. Where is the interruption? And as these arrows here indicate, an interruption here, or an interruption here, an interruption here, or an interruption here, all would lead to a Horner's syndrome. And so when reading a question that describes a Horner's syndrome, you have to continue, once you've identified that there's a sympathetic loss to the head, you need to keep seeing what other clinical findings are associated with that finding in this patient to help you localize where the lesion is. Because depending upon the level of the lesion, there will be other losses, often somatic losses associated with it, and that will help you localize the lesion. Also take note, and we won't go into this in any detail now because we'll reserve that for our discussion of the central nervous system in neuroscience, but notice that even though the preganglionics are starting from the spinal cord levels T1 to L2, they in turn are controlled by descending fibers that come from the hypothalamus, that come through the brainstem down the cervical spinal cord, and are the upper motor neurons that innervate these lower motor neurons that are the preganglionics. So if we interrupt those, what are called the descending hypothalamic fibers, that would also lead to a Horner's syndrome. So you can have a central nervous system lesion at brainstem or cervical levels, or you can have a peripheral nervous system lesion anywhere from T1 on up through this pathway, all of which could lead to a Horner's syndrome. So that concludes our discussion of the sympathetic nervous system. The parasympathetic nervous system is much simpler to deal with because we don't have the same fundamental issue. We don't have to distribute all over the body. Parasympathetics do not go to the body wall. They don't go to the chest or abdominal wall. They don't go to the limbs. They only innervate visceral organs in the thorax, abdomen, and pelvis, as well as a few visceral organs in the head. So we know that the preganglionics come from cranial levels and from sacral levels. At cranial levels, they're in the brainstem associated with cranial nerves 3, 7, 9, and 10. At sacral levels, associated with S2, 3, and 4. Let's discuss these pathways in two separate groups. For the first group, what we'll do is we'll combine the sacral outflow along with the vagus outflow. So we'll combine vagus plus the sacral 2, 3, and 4 parasympathetic outflows. I'm combining them together because their pathways are fundamentally the same. Remember we said that parasympathetics have their postganglionic cell bodies close to their end organs. They're so close to their end organs that they're actually in the wall of the organ. Those groups of postganglionics that are in walls of organs are given the name terminal ganglia. So here are terminal ganglia, here are terminal ganglia. They're in the walls of the organ. So if the end organ that's being innervated is, let's say, a smooth muscle cell in the wall of the stomach, the cell body that innervates that is in the wall of the stomach, in that terminal ganglion in the wall of the stomach. So that's true for the vagus. It's true for S2, 3, and 4. The only thing that's different is what regions are these end organs in. And fundamentally, we can say that the organs that are in the thorax and fundamentally the upper portion of the abdomen get their parasympathetics from the vagus, and those that are in the lower portion of the abdomen and the pelvis get their parasympathetics from the sacrals. And that's all there is to it. The remaining parasympathetics, 3, 7, and 9, their responsibility is to innervate structures that are in the head. They do it in a slightly different way. Instead of having terminal ganglia, in other words, cell bodies in the walls of the organs, in the head we have 4 discrete parasympathetic ganglia: ciliary ganglion, submandibular ganglion, pterygopalatine ganglion, and otic ganglion. And here the relationship is very simple. All of the preganglionics in the third nerve synapse in the ciliary ganglion, all of the preganglionics in the ninth nerve synapse in the otic ganglion, and all of the preganglionics in the seventh nerve synapse in either the submandibular or pterygopalatine ganglion. So that takes care of the preganglionics. The postganglionics, the ciliary ganglion takes care of 2 end organs, 2 smooth muscles in the eyeball, sphincter pupillae and ciliary muscle. Postganglionics of the otic ganglion take care of 1 salivary gland, the parotid gland. Postganglionics in the submandibular ganglion take care of 2 salivary glands, submandibular and sublingual gland. And that leaves the pterygopalatine ganglion taking care of everything else in the head that gets parasympathetic innervation, which would include the lacrimal gland, as well as all of the glands, serous and mucous glands, of the mucosal surfaces in the head: nasal mucosa, oral mucosa, paranasal sinus mucosa, pharyngeal mucosa. All of those mucosal glands get their innervation from the pterygopalatine along with the lacrimal gland. So that concludes parasympathetics. 3, 7, and 9 use the parasympathetic ganglia of the head. 10 and the sacral parasympathetics use the terminal ganglia in the walls of organs. So that concludes our discussion of the autonomic nervous system.